Howdy, my name is Jaslyn Gonzalez, and Natalie, Heaven, and I will be presenting over our Project 5. Chapter 7, Humoral Immune Response. First, we're going to talk about the phases and types of immune responses. Humoral immune responses consist of multiple phases, each playing a role in antibody-mediated immunity. In antigen recognition, we start off with naive B cells that are expressing membrane-bound immunoglobulins like IgM and IgD, which encounter and bind to a microbe. Once activated, these B cells will undergo clonal expansion, leading to proliferation followed by differentiation. In differentiation, B cells mature into plasma cells specialized for antibody secretion, undergo isotope switching or affinity maturation, and/or they become memory B cells. Humoral immune responses can also be classified into T-dependent and T-independent, depending on whether B cell activation requires helper T cells. T-dependent responses do require T helper cells, which is what leads to isotype switching, affinity maturation, and long-lasting immune memory. T-independent responses are triggered by non-protein antigens like polysaccharides and lipids, which leads to the limited antibody response being composed of low-affinity IgM. The primary immune response occurring at initial antigen exposure is relatively slow, lasting 5 to 10 days, smaller peak response, and usually produces IgM antibodies. There is lower average affinity as well. The secondary response is relatively faster, lasting 1 to 3 days due to memory B cells, and has a larger peak response. We will also see heavy chain isotype switching. This immune response is the basis for long-term immunity and vaccination. Stimulation of B cells by antigen, T-dependent antigens. They're protein antigens that require interaction with CD4 helper T cells to fully activate B cells. Via BCRs, B cells will recognize and bind to an antigen. It will then be presented on MHC class II molecules to helper T cells. Activated CD4 plus T helper cells will then recognize these MHC2 antigens and provide essential activation signals. CD40-CD40L interaction is where helper T cells express CD40L, which binds to CD40 on B cells. This promotes proliferation and survival. Cytokine release is where helper T cells secrete cytokines, which direct B cell differentiation and antibody production. Lastly, B cells undergo isotype switching depending on cytokine signaling, affinity maturation to enhance the affinity of antibodies for their antigen, and generate memory B cells for a faster and stronger immune response upon re-exposure to the same antigen. This response is critical for vaccine development and long-term immunity. Stimulation of B cells by antigen, T-independent. These are non-protein antigens like polysaccharides and lipids which activate B cells without the help of T cells. These antigens have repetitive epitopes, which allows for direct BCR cross-linking and string activation. CR2, being attached to a complex of proteins, are involved in delivering the activating signals to the B cell. This B cell will recognize a microbial antigen through Ig receptors and bind to C3d by the CR2 receptor. This is known as complement activation. This response primarily produces IgM antibodies, has no affinity maturation, and has minimal memory B cell formation, leading to a short-lived immunity. These responses provide rapid, early protection against infections.
Role of innate immune signals in B cell activation. The innate immune system is able to stimulate B cell activation. This is possible via the complement system. Once a microbe is coated with C3d protein, CR2 receptors on B cell surfaces will recognize the C3d simultaneously to the neighboring BCRs, recognizing the microbial antigen. CD18 and CD81 that are part of the protein complex with CR2 would deliver the necessary activation signals. Another innate immune signal in B cell activation are TORs on B cell surfaces. that recognize PAMPs or pathogen-associated molecular pattern of a microbial antigen. This recognition simultaneously occurs as microbial antigens are being recognized by BCRs. The TOR recognition and activation of B cell is shown to the far right, and the complement activation of B cells is shown to the left, and the left part of the image to the right. Both of these innate signals contribute as co-receptor or secondary signals that do not require T cells for B cell activation, consequently making these activation pathways, as mentioned with the CR2 co-receptor and the TOR receptor, as T-independent. Heavy chain isotype class switching. Heavy chain isotype class switching refers to the Fc region of an immunoglobulin switching to represent other Fc classes. The Fc region is the stem-like part of the immunoglobulin, and this elicits the respective antibody function for the immunoglobulin. Helper T cells will stimulate IgM and IgD on B cells to produce antibodies with different heavy chain isotypes. The isotype switching is induced by the CD40 ligand, and we can see this represented on the image to the right, and cytokines. These cytokines are produced by the follicular helper T cells, which determine the type of class or isotype of an antibody. And we see this being demonstrated in the image to the right as mentioned. And we can also see that interleukin-5 stimulates the activated plasma B cell to produce IgA, and interleukin-4 stimulates to produce or class switch to IgE and IgG. And to the left, we have the types of antibodies that are produced in our system. Affinity maturation. Affinity maturation occurs on cells that are stimulated by T helper cells, T-dependent, making this process T-dependent. This process consists of affinity of the antibodies increasing as a response to prolonged and repeated exposure to a specific protein antigen. Affinity maturation occurs in the germinal centers of lymphoid follicles, as shown in this illustration. The antibody's affinity to a protein-specific antigen is increased due to somatic hypermutation in the Ig genes. The Ig genes are not shown here, but the process in which B cells are selected is demonstrated. B cells with higher affinity antibodies are selected, and those that are unable to develop high-affinity antibodies and do not pick up antigenic proteins from FDCs are unable to present the proteins to receive survival signals from T helper cells. B cells that have higher affinity present their antigenic proteins to helper T cells and receive their survival signals, creating memory and plasma cells.
Regulation of humoral immune responses, antibody feedback. As the natural decline of the immune response begins, activated B cells undergo apoptosis. However, when they are produced, the secreted IgG antibodies circulate and bind to any remaining antigen, often forming immune complexes or antigen-antibody clusters. B cells specific to that antigen interact with the secreted IgG and its antigen-antibody complex, and the IgG then binds to the antigen part with the membrane-bound Ig, and its Fc region binds to an Fc receptor, known as Fc gamma RIIB, on the B cell. This receptor sends inhibitory signals to the B cell, preventing further activation and stopping antibody production via the immunoreceptor tyrosine-based inhibition motif, or ITIM. This process is known as antibody feedback, which regulates B cell activation once the antigen has decreased and ensures that no autoimmune response is developed against self-antigens. Effective functions of antibodies. Antibodies are secreted when B cells are activated. They are able to perform different functions depending on their isotype. Functions of the antibodies include neutralization, opsonization, phagocytosis, antibody-dependent cellular cytotoxicity, IgE cell-mediated reactions, and the activation of the complement system. Antibodies, mainly IgM and IgG, can neutralize antigens in various ways. At epithelial surfaces, they block the entry of microbes. At other times, they prevent microbes from binding to cells and infecting them. Toxins produced by microbes can also be neutralized by antibodies. These actions help neutralize extracellular microbes. The classes of IgG enhance phagocytosis by opsonizing a microbe. This is done when the opsonized microbe binds to the phagocyte's Fc receptors, which then stimulate a signal to engulf and kill the microbe. Different Fc receptors stimulate different responses. ADCC begins with an IgG binding to a host cell. The Fc region is then recognized by Fc gamma RIII on natural killer cells, which activates the killing of the antibody-coated cell, similar to opsonization. An IgE and eosinophil mast cell mediated reactions. IgE can activate eosinophils or mast cells to help defend against parasitic infections or allergic diseases. When IgE binds to helminths, it recruits eosinophils. Th2 cells also assist in the eosinophils in enhancing their defense functions through the secretion of IL-5. The eosinophil and IgE then degranulate to kill the helminth. The complement system's classical pathway is activated when antibodies, usually IgM or IgG, recognize an antigen. Once activated, all three pathways involve the enzymatic activity of complement proteins that generate effector molecules to help eliminate the microbe, such as C3A, C3B, C5A, and C6 through 9. C3A and C5A lead to inflammation, while opsonization occurs through C3B. Cell lysis is ultimately achieved through C6 through 9, which forms the membrane attack complex. During the complement system's activation, a second signal that activates B cells occurs when a microbe binds to C3D. The B cell is able to recognize the microbe bound to the C3D, while the CR2 receptor recognizes the C3D. The CR2 receptor is crucial because it acts as a second signal to activate the B cell. Vaccines. Vaccines aid in stimulating an immune response against microbes by exposing the body to non-pathogenic forms. There are a total of 7 different types of vaccines, with mRNA being the newest form, developed for the COVID-19 pandemic. One of the most effective types is attenuated vaccines, in which the microbe, whether a virus or bacteria, is weakened just enough to prevent the severity of the disease while still producing an immune response. Unfortunately, they are not recommended for those with immunodeficiencies or possibly pregnant individuals. A subunit vaccine contains parts of a purified microbe that are needed to elicit a response. These parts can be proteins or polysaccharides that make up the microbe. A conjugate vaccine is made of polysaccharides from a microbe that require additional help, so they are coupled with proteins that can stimulate a helper T cell response. Unlike other vaccines that can be cultured, synthetic vaccines consist of proteins made in a lab. They often elicit an immune response more quickly. Viral vectors involve inserting a microbe into a harmless virus to generate antigens and infect host cells, thereby triggering an immune response. Additionally, an antigen's gene can be injected into the body to be recognized by APCs, which is known as a DNA vaccine.